Thank you for the invitation and for the kind introduction. It's the workshop is certainly very welcome. I think it's a very good initiative to do a Metabolomics India workshop, and really happy to participate in this. I've been listening all the talks since early afternoon. We would like to present some of our work on metabolomics. So my lab primarily worked on microbial metabolomics. We started working on metabolomics 12 to 15 years ago. And at that time, we didn't call it metabolomics. So so our background is more from 13C metabolic flux analysis. So we would take bacteria, grow them, feed 13C isotopic label, try and trace the 13C isotope in different metabolites. The concern there was to be able to accurately annotate and quantify the metabolites and the 13C uptake in those metabolites. And therefore, we are very concerned about accuracy of annotation as well as the quantitation. Now, fast forward 10 years later, we are working on human metabolomics. And so this is a bit of a diversification compared to what we were doing a bit earlier. So in the human metabolomics, our objective is primarily developing new diagnostic tests based on mass spectrometry and artificial intelligence. And again, two broad areas. One is precision diagnostics, and that's the work that I'm going to present today. And our current focus is on diabetes. So predisposition, disease severity, and prognosis. The other area that we're interested in is sports medicine, optimization of human performance, longevity. The cartoon shown on the right-hand side is of mitochondria, the small organelle that are present in our cells, and they are the sites for fat oxidation and other energy metabolism. So fat oxidation goes through beta oxidation, which creates intermediates, and those intermediates, especially the acylcarnitines, are visible and they can be seen in our blood samples. And they give you an idea of the mitochondrial fitness, the fueling pathways, and so on. And this can provide personalized guidance on training and nutrition. A slightly different area of work is spent media analysis and genome scale metabolic modeling in my lab. I will not be talking about that area today. So the topic is on type 2 diabetes. We all know really large number of diabetic population all over the world, in India as well. It's a progressive disease starting with high levels of blood glucose, which eventually lead to complications. And so a significant fraction of patients develop nephropathy, retinopathy, and so on. And this was also identified as an important comorbidity for COVID deaths. And the objective is early identification. Intervention can prevent or delay its progression. So obviously, we as a commoner, we know that diabetes is one disease. And of course, those who are more aware, they know that there is type 1 diabetes and type 2 diabetes. But recent literature shows that diabetes, there may be different types of diabetes. In cluster 1 is more what we commonly know as type 1 diabetes, whereas cluster 2 to 5 are what are now we know as type 2 diabetes. There are variations. Some of the diabetes are more aggressive. They lead to complications much faster within 5 to 10 years of diagnosis of diabetes itself. Whereas some others are more not so aggressive. The progression is slow. The complications are less likely. So, for example, the mild, obesity-related diabetes, the mild age-related diabetes, these are somewhat less aggressive compared to the others. And therefore, now diabetologists have started to recognize these clusters and determine the therapies based on the clusters. This is the Indian study, recent publication from an Indian group, where the type 2 diabetes again has been broken into 4 types, subtypes, And we can see what's the percentage, 26% for severe insulin deficient, all the way mild age-related, 35% or so. And as I mentioned, the mild age-related diabetes, which with the onset at later age, is the least aggressive of all. So it's important to find out if what class of diabetes and patient stratification. Current biomarkers are glucose and HbA1c. So HbA1c is also an indication of glucose control or glycemic control. And therefore, these biomarkers do not give you an indication of the progression of disease or severity of the disease, how much damage it has already caused. Importantly, diabetes as well as the complications are initially clinically silent, and therefore there are no symptoms. Once the symptom starts, the disease has already progressed significantly. The key questions that we like to look at in our research, first of all, what is a good biomarker? This is an important question. We know that glucose is a good biomarker, and there are many other biomarkers that are currently used in clinical practice. And there are some biomarkers that are reported in literature studies. What is the diagnostic yield of a biomarker? Can it provide you actionable results, or it is just a value that may not have much significance? Then the next question we are asking is, can we use a signature of metabolites as a biomarker, as opposed to looking at just one or two biomarkers at a time? And then finally, can we look at patient stratification and identifying those at risk of complications? So our current study is in collaboration with Dr. Rakesh Sahai and Dr. Manisha Sahai, where we have taken a cohort comprising of 4 groups: healthy, type 2 diabetic with kidney disorder, type 2 diabetic with cardiovascular disorder. Plasma samples were taken, and LCMS and GCMS was performed. Just to give you a brief of where we would want to use LCMS versus GCMS. So GCMS, as the name suggests, it's gas chromatography. So it can be used for anything that is either volatile or can be converted into volatile compounds. So via derivatization. So we can convert amino acids, sugars, sugar acids, alcohols, etc. into volatile compounds by standard derivatization processes. And we use GCMS for those. GCMS also provides advantages in being able to separate isomers. Leucine, isoleucine can be separated. Various C6 sugars can be separated, and so on. LCMS, on the other hand, can be used for compounds that are not volatile, bile acids, acylcarnitines, and so on. And in fact, with LCMS untargeted methods, we can easily see 500 to 600 compounds. The mass spectrometry methods can also detect ppb or ppt level of concentrations. And just to put things in perspective, glucose in blood plasma is 80 milligram per deciliter, which is literally 800 ppm, which is a very high concentration. Our workflow has 2 parts to it. The upper part is untargeted metabolomics, where I have shown MS and untargeted annotation of compounds, relative quantification of compounds on the right-hand side. The lower part is a targeted metabolomics. So once I already know what are the metabolites I'm looking at, then we want to develop more targeted methods. The mass spectrometer used in the targeted method is triple quad versus high resolution mass spectrometers such as TOF or Orbitrap are used in the upper path, which is for untargeted metabolomics. So we work on both these sort of pathways. The untargeted pathway leads to 400 to 500 metabolites, typically in blood plasma. In targeted methods, we can easily develop method for anywhere between 50 to 100 metabolites in one single run, in blood plasma. In untargeted metabolomics, the key challenge is data analysis. So we have our own software, which takes MZML files from mass spectrometer and does peak picking, annotation, removal of noise, removal of in-source fragments, isotopes, adducts, and so on, and then removes the redundant features. So, and then this is a fully automated software which which does all this using various algorithms. As a first step, we would like to see how the quality of the data is. So for this shows is the total ion chromatogram or TIC, and this is shown for the QC files. So begin with, okay, this looks good. Then I go and see whether my extracted ion chromatogram looks good. This is the extracted ion chromatogram. The software has already identified the peak boundaries, and we see now that okay, the peak is of this width, the peak height is so much, and so on. Then we want to also see how is the mass detector working, and what is the mass shift, what should be the mass tolerance that I should use. So this is intensity versus M by Z in a profile mode of the mass scan. And if I go to a typical data that is generated from a high resolution mass spectrometer, you can see that the width of the peak, which I can sort of divide into half, that I would take as the mass error. Here I have taken this as 10 ppm, or it turns out to be 10 ppm. And based on such analysis, which you can do using our software, you can decide what should be the so obviously, if you use lesser mass error, you are going to ignore certain data. If you use much larger mass error, you are going to start capturing noise into your data analysis. The next step is peak alignment. So the left hand side is before alignment and right hand side is after alignment. So this is an important step. This has to be done using certain algorithms between two files, between a reference list of peaks versus the peaks in a file, and so on. Annotation is an important part. So we do noise removal in the MS2 data. So this is something which is not done very routinely in most of the softwares. And therefore, as you can see, the confidence in these annotations is very high. So we remove noise both in the experimental data, which is the upper graph, and the library data, which is the lower graph. And then we get really high quality annotations, which would pass the test of a human eye. The human expert would certainly, tick this as a good annotation. Then we need to look at batch effects. So this is something which has not been discussed in the workshop today. So we can the mass spectrometer detectors are notoriously non-quantitative. So there is a drift in the intensities, and therefore you will see batch effects. The samples that are injected on day 1 versus day 10, there will be batch effects. So we need to remove these batch effects. Certain kind of normalization has to be done. So that is done. And then we want to see the peak quality and the peaks and so on, and that can also be done. So these are the features that we have seen. These are the metabolites that get annotated, over 300 metabolites, all kinds of sugars, purines, pyrimidines, organic acids, derivatives, vitamins, and so on. And as I mentioned, acylcarnitines is another thing. Now, looking at the data, as we start clustering the data using the metabolomics data, so we start clustering the samples using, so these are the samples, the dots are the samples, which are non-diabetic, the plasma sample from non-diabetic volunteers. And the crosses are the plasma samples from diabetic volunteers. And as we can see, there are already two groups, even in this small population, there are two groups of diabetics. One group is closer to being healthy, and another group, we know that it is very different from the healthy population. And the other study we have done where the diabetic kidney disorder cohort was there. So this group is actually moving toward complications. So I can already perform patient stratification using these kinds of metabolites. And I'll show you what are the metabolites that will. So this is a metabolite 1,5-anhydroglucitol, which gives you an idea of glycemic control over the last 15 days or so. And as you can see, the 1,5-anhydroglycerol level should be high. They are high in healthy, but in diabetic population, in half of them, they are quite low, which means that this population has got poor glycemic control, at least in the last 15 days. Fructose is a C6 sugar, which can potentially accumulate in our blood plasma. And we can see that in diabetes, it accumulates much more than in the healthy population. And fructose has been reported to be potentially more harmful in terms of its effects on cardiovascular disease and so on, microvascular damage, than glucose itself. So this is something to watch out for. Then another thing to watch out for is arachidonic acid, which is an inflammation marker. We can see that it is elevated among the diabetic population, much more than the healthy population. In terms of the complications, if I look at the kidney disorder as a complication, then there are certain uremic solutes or uremic toxins that are known. And we can see that phenylacetylglutamine, which is one of the well-known uremic toxins, that has low levels in healthy population, relatively high levels in the DKD population. But in diabetic population also, there are already few patients which start showing elevated levels, which may indicate that there are certain complications that have started to set in. Indoxyl sulfate is another uremic solute. Again, in DKD, the kidney disorder, the levels are much higher than the healthy population. And in diabetic population, there are few people who have relatively higher levels. So looking at the box plot, so the left, in each graph, there are three boxes. So first graph is non-diabetic, second graph is diabetic, and third graph is those with kidney disorder. So creatinine, which is measured from LCMS, the same assay, is higher in the kidney disorder people. Obviously, the kidney disorder is diagnosed based on creatinine levels, so it is expected to be higher. Phenol sulfate, which is a known biomarker for kidney disorder, goes up from non-diabetic to diabetic and there is a progression in the kidney disorder. And we find few other biomarkers which also progressively go up from non-diabetic to diabetic to kidney disorder. So ribothiamidine is one example, arabitol is another example. And we have few other examples that that also show a similar pattern. So just a very quick introduction to what we do with spent media analysis. So this is a fermenter. We perform exactly same type of LCMS based metabolomics on the fermenter samples. Use that data to fit to what is called as a genome scale metabolic model. And the use case here is to optimize the fermentation process. So just to give you an idea, these are the data sets that we have collected for some fermentation. Red is high and blue is low. So every little row here is one metabolite, and every column is one time point. So going from 0 time point all the way up to 48 hours, you can see that there are certain metabolites from the media are getting consumed, and there are certain metabolites that are getting secreted into the media. So we have seen this for various systems. The data here is shown for E. coli, and we do it for E. coli, CHO cell culture, and so on. And we do it with some collaborators also as well for other systems. So we see that every culture, every fermentation has a hierarchy of substrate utilization. Some substrates are utilized early, some are utilized later. And it also, every culture also secretes certain metabolites into the fermentation broth. And that's something that we use as a, to model the metabolic model and use it for optimization purposes. So in summary, we know that data analysis is a key step in metabolomics, especially the high resolution TOF type of LCMS data or Orbitrap type of LCMS data. And the software that we have provides several advantages. It is available on the web. Number of metabolites get perturbed in diabetes and in complications, and they can be potentially used as biomarkers for progression of disease. The data that we have is based on relatively small population, that needs to be expanded. We need to collect data on relatively large populations and hopefully develop diagnostic tests in the and this data has to be on different cohorts based on ethnicity, age, gender, and so on. Of course, this kind of work requires a lot of funding. Our primary funding source is DBT, Department of Biotechnology, Government of India. We also have funding from L&T, the donation funding from L&T, Praj Industries, Sigma SGS, Wadhwani Foundation, Koita Center. And in the diabetes project, our primary collaborators are Dr. Rakesh Sahai, Dr. Manisha Sahai, Dr. Gurjar at IIT Bombay, and more doctors are joining us in that project. In the fitness space, we have been collaborating with Dr. Marcus Rani, Dr. Ashish Contractor, Mr. Venkat, who organizes and helps the marathon runners. And thank you so much for your attention. I'm happy to answer any questions.